Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, we are from group 5 and we will present our presentations about Plantibodies. But first, let us introduce ourselves. My name is Nadia Nura and my student number is 163.
My name is Muhammad Faryasam with NIM 164.
I am Zagi Raza Nur Asyam with NIM 167.
I am Anissa Erkawati with student number 168.
Without any further ado, let's get started. Okay, let's start with introduction. Plants, of course, don't have an immune system like humans and animals. They don't produce antibodies. In the early 1990s, transgenic plants were produced genetically engineered to incorporate genome and express foreign genes, such as mouse genes that produce antibodies against pathogens. Jump to the topic discussion, we will discuss about antibodies and the antibody structure. The first one is plant antibodies. What is antibodies? Antibodies are glycoproteins produced by the adaptive immune system in humans and other vertebrates. They recognize and bind particular target antigens with great affinity and specificity, allowing them to clear pathogens and other unwanted material from the body. Also, antibodies can be used for the immunomodulation of endogenous targets, e.g., inhibiting the activity of a host enzyme or metabolite that may be involved in the infection process. Functional antibodies produced by plants against specific plant pathogens that have been shown to increase the resistance of the host plant to that pathogens include the following. Plantibodies to tobacco mosaic virus. In tobacco decrease infectivity of the virus by 90%. To beat necrotic yellow vein virus, also in tobacco, provides a partial protection against the virus in the early stages of infections and against development of symptoms later on.
Genetic engineering and biotechnology now offer additional opportunities for disease prevention because they allow disease resistance genes to be introduced into plants from diverse sources with none of the disease barriers that apply to conventional strategies. The topic number 2 is antibody structure. Monovalent single-chain variable fragments compress the variable regions of the XC and LC, joined by a short linker peptide that is usually rich in glycan and serine. These have the least stringent requirement for folding and assembly, and also penetrate tissues effectively because of their small size. This makes them suitable for expression in diverse environments. Antibodies exist both as soluble proteins and as membrane-bound receptors on the surface of B lymphocytes. Their primary function is antigen binding, and this often has a direct inhibitory effect on the pathogen target. For example, the neutralization of a bacterial toxin or the prevention of viral attachment to host cell.
Next, about antibody structure. Antibodies are remarkably diverse and have the capacity to recognize tens of millions of different antigens in the environment. This diversity is achieved by somatic gene rearrangement, reflecting the fact that germ-line immunoglobulin gene exist as multiple segments that are combined in different ways in different B cells. Interaction between a single epitope and its corresponding paratope is known as antibody affinity, whereas the combined strength with which a multivalent antibody binds a multivalent antigen is the antibody affinity. Many antibodies will bind only to native antigen or fragment that have folded properly to form multiple nonlinear interaction between the antigen and antibody. Full-size antibodies are usually multivalent, enabling them to bind to their target with a high affinity, whereas fragments might be multivalent or univalent depending on their construction.
Next, transgenic plants producing antibodies against the pathogen. Plants lack an antibody-making machinery, but as mentioned elsewhere, DNA technology has made it possible to transform plants with additional genes that make possible the production of functional recombinant antibodies. Such plants produce antibodies or Plantibodies can be full antibodies molecule, fragments, or single-chain Fv fragments produced against specific plant virus, and they are targeted against the same virus. They have been fully expressed in leaf and seed of some plants and accumulate in intercellular space, chloroplast, and the lumen of the endoplasmic reticulum. Single-chain Fv fragments are also expressed in intracellular space, which allow them to be more effective in improving the resistance of the plants against the specific virus. Next, and the conclusion are: 1. Plants do not naturally produce antibodies. They can be engineered to do so by introducing the corresponding hemoglobin genes. Number 2. Antibodies exist both as soluble proteins. Number 3. Antibodies or Plantibodies can be full antibody molecule, fragments, or single-chain Fv fragments produced against specific plant virus, and they are targeted against the same virus. That's all. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.